Good day everyone. In current lecture we'll continue to discuss enterobacteria. Topic of that lecture is Salmonella. So again, as any other enterobacteria, it is gram-negative, roche bacteria, facultative anaerobic non-fastidious. Salmonella is non-capsulated, non-spore-forming, and highly motile by peritrichous flagella. It is, as any other bacteria, again, it is parasite of the intestine. And nowadays, more than 2,500 serotypes are described, and main disorder caused by Salmonella is known as a salmonellosis, or it is a type of food poisoning, food infection, and unique disorder caused by Salmonella is known as enteric fever or typhoid. And bacteria have number of antigens. It is surface O antigen, flagellar H antigen, and as well it have VI antigen, which is an analog of a K antigen in other bacteria, but in Salmonella it is known as a VI antigen, just unique name. So again, if we talk about factors of virulence or pathogenicity, bacteria have lipopolysaccharide O antigen, which can reduce binding of antibodies to the surface of bacteria. It is a protection against phagocytosis, and when released into the systemic circulation, it have non-specific toxic effect. Bacteria similar to Shigella have no capsule, but it is surrounded by a slim layer, which is an analog of a capsule, thin mucoid layer. In the case of Salmonella, it is again known as VI antigen. Functions are the same: protection against phagocytes and complement. As well, it can help to stick to surface of enterocytes and helps bacteria to estimate infection in GIT. Bacteria is highly motile and have multiple flagella, which are acting as H antigen. As well, those flagella helps to invade in the epithelium. It helps to make more harder movements between intestinal cells. It is covered with the fimbriae, which helps to fix to the surface of epithelium. It can act as a colonizing factor. As well, Salmonella can produce biofilms on the surface of some tissues, and again it's possible partially thanks to that fimbriae. Bacteria have several factors of adhesion and invasion. We can combine them as a surface associated proteins. They help to establish an infection in GIT. And one unique enzyme, which is known as a superoxide dismutase, it protects against different enzymes and protective systems based on rectified oxygen. Again, the main scheme of differentiation of Salmonella is serological scheme. It is a serotyping, and that scheme is known as a Kaufman-White. It is based on the structure of lipopolysaccharide flagellar antigens, which exist in two forms, H1, H2, and that slim layer VI antigen. In general, nowadays we have more than 60 serogroups described, and again about 2,500 serotypes or serovars. So, in correct way, classification of Salmonella is based on antigenic properties. We have only two species of Salmonella: Salmonella bongoria and Salmonella enterica. Salmonella enterica is subclassified into 6 subspecies. Salmonella enterica subspecies enterica is the most important, and Salmonella enterica subspecies enterica is further classified into serotypes, and that serotype Salmonella typhi, which is mainly just named as a Salmonella typhi. In correct way, should be named as a Salmonella enterica subspecies enterica serotype typhi, SEE typhi. But of course, it is long; it's not very useful to use that name, and usually we abbreviate it till Salmonella typhi, but again it's not a separate species, it is a serotype of Salmonella enterica. And in general we have huge number of different serotypes. They are named by the disease they can cause, or by geographic region they were first identified, or by type of animal which is affected by this bacteria. Again, enterobacteria routinely use culture medias McConkey agar. Salmonella and Shigella are lactose negative, so colonies of Salmonella McConkey agar will be pale, decolorized. Agar is not changed. Colonies are small. Bacteria is less fast growing than other enterobacteria. The same selective culture media as was used for Shigella, XLD agar, SS agar, and HE agar, but in the contrast with Shigella, you can see that Salmonella will give you a very bright result. It is a black color of colonies. XLD agar will be very bright pink, SS agar bright yellow, and HE agar will be less colorful. It is pale, but colonies are intense, dark green or black. In any culture media, Salmonella is easier identified with from Shigella, and it's obvious which microorganism do you have in your current case. If we will use Kliegler iron agar, then we have very diverse properties because Salmonella have huge number of different serotypes. Results of Kliegler agar can be absolutely different. Some Shigella produce gas, some produce H2S, but in general fermentation of glucose always takes place with formation of acid and gas. So we have actually two different diseases, typhoid fever and paratyphoid fever, but the only difference is the agent which is causing the disease. Symptoms, pathogenesis, everything is the same. So very often when especially etiology of disease is not confirmed, the common name used for both typhoid paratyphoid fever is enteric fever. It means that any bacteria not confirmed which one causing the disease. As for any GIT disorder, diarrheal disease, enteric fever is transmitted by fecal-oral route through contaminated food products or water, and transmitted from human to human or from animal to human according to which bacteria is present in your case. Incubation period can last up to 3 weeks and about a week of prodrome stage. In general, up to a month of asymptomatic periods is possible, and main course takes about a week or little more. Manifest with the headache, malaise, loss of appetite in progress of the disease. Symptoms of toxic shock will appear as a pale skin, decreased heart rate, and in severe cases retardation with the fever and hepatosplenomegaly are possible. Unique symptom of enteric fever is a coated tongue with teeth prints on the surface, and in some cases roseolar rashes can appear on the skin. It's easier to differentiate with another because when you will press on the element of the rashes, it will disappear, become pale, but when you will remove your finger, it will appear again. If liver and spleen are affected seriously, then jaundice can appear with yellowish skin and sclera, and decolorized stool and beer-like urine are possible as well. So pathogenesis of salmonellosis is very similar to Shigella. The same invasion through M cells. Macrophages are not involved in that case, but M cells will be damaged much more seriously, and actually destroyed M cell will let Salmonella to invade into the laminar space easier. So bacteria will invade epithelium through damaging of the cell. Again, it have a lot of adhesion factors which help to stick to the surface of epithelium, and when M cells are affected, bacteria will invade into the epithelial cells, increase recruitment of lymphocytes and mononuclear cells. In general, it will lead to proliferation of regional Peyer's patches, and with the time it can lead to necrotizing of Peyer's patches of intestinal cell wall and lead to ulcerations. When lymph nodes will be damaged seriously, bacteria can easily invade into the systemic lymph and blood circulation via Piquet duct, and usually that stage is called as a primary or asymptomatic bacteremia because systemic symptoms will not present. Bacteria will shed in few amount in the bloodstream. When bacteria will spread via blood circulation through the whole body, usually it will affect regional or tissue macrophages and blood macrophages monocytes, where Salmonella will multiply intracellular. Previously, those cells were named as the reticuloendothelial system. You can make that definition as well. but after multiplication in tissue macrophages, bacteria will again invade into the bloodstream, but now in much more higher amount, and it will lead to secondary bacteremia, which can last up to several weeks and will manifest with common symptoms of toxic shock, because bacteria in bloodstream will be destroyed and lipopolysaccharide will be released into the systemic circulation directly, induces severe symptoms, release of cytokines, and multiple damage of organs and tissues. Actually, any organ can be affected. That's why we have multiple clinical manifestations, but again, mainly it is liver and spleen with focal necrosis and hemorrhages in tissues in pathologic investigation. So without diagnosis, it is very possible to have lethal outcome because of formation of ulcers, perforation of intestine with the peritonitis, internal bleeding, intestinal bleeding, and lethal outcome is rare, but state can be very severe and disease can last for months. So if diagnosis will be performed in correct manner and as soon as possible, then mainly you will observe fast decrease of symptoms, fever will decrease, general state will improve, person will become normal in weight, normal in general activity, and recovery is complete. In rare cases, about 10% it is possible to have relapse of disease if treatment was incomplete, or self-convalescence can lead to relapses of the disease again. So in that case, of course, it is important to prescribe antibiotics as soon as possible. It is systemic infection with invasion of bacteria in the blood circulation, and of course it is essential to kill bacteria completely. So the same ampicillins or cephalosporins should be used as the primary therapy, but again, nowadays antimicrobial resistance is high and adjustment of therapy is required according to susceptibility testing or to endemic data. The same non-specific measures of prophylaxis should be followed. It is isolation of ill person, identification of the source of infection, eradication of carriage people, and of course proper sanitation, proper hygienic measures are the most important to prevention of any enteric disorder, especially enteric fever. So again, we said that mortality rate is kind of low, but in common worldwide, about 25 million of people are suffering from the disease annually. And of course, even that some one person is 200,000 deaths annually, it is very significant number. And in those pictures, roseola rashes and coated tongue, teeth prints are not very good. It's difficult to actually identify diseases, rarely photoed and registered, but those symptoms are kind of unique for typhoid. Another group of disease which is caused by Salmonella is known as salmonellosis. Any other Salmonella except typhi group can cause that disorder. Usually disease is transmitted from animals. It is a bird, poultry, very rarely human-to-human transmission were registered. Way of transmission is fecal-oral through milk, meat, eggs, or other products obtained from the birds, or some use of water and soil in vegetables which are contaminated with water, or some fecal matter of those animals. And again, it can be eliminated through some food products when they are not washed properly. Incubation period is very short. Usually first symptoms will appear after 6 to 8 hours after ingestion of the product. Pathogenesis is the same, but here bacteria will produce toxin which resembles cholera toxin. It will affect exchange of electrolytes in blood and water, and for about 5 days people usually have nausea, vomiting, and diarrhea with mucus, but without blood in feces. When bacteria will again invade the bloodstream and release endotoxin, fever and common symptoms are possible: weakness, tiredness, malaise with abdominal pain, but usually in salmonellosis it is rare. So convalescence is spontaneous in about a week. In severe cases it is possible to have renal failure or some bacteremia, but in routine salmonellosis those complications are rare. Common treatment and preventive measure again are applied here, and especially if it is some bird products, then of course it is better to buy known sources in markets when quality of what products is not controlled by government. It's better to avoid food products, or if you will buy, then good thermal cooking should be performed. Fresh food products are dangerous for consumption. If we'll talk about immunobiological preparations again, we have bacteriophage. It's almost the same as in any other case, but in the case of Salmonella, it is important to have it, especially when kids are suffering, when antibiotics are contraindicated or choice is very limited. We don't have officially registered international bacteriophages, but in Russia we have bacteriophage for different groups of bacteria, serogroups. It is solution for oral intake. It is used for treatment when disease is already present, and it helps to release symptoms and to kill bacteria much faster. In the case of enteric fever, it's better to prevent because we have a large number of vaccines which are registered and approved by CDC and WHO. So according to CDC instructions, we have two types of vaccines: inactivated typhoid vaccine, conjugate based on VI antigen. It is known as TCV vaccine. It may be given to people older than 2 years of age, and again the main indication is traveling or trip to endemic area, or people who are working with those people, main doctors and medical staff, or laboratory workers. Second type of vaccine is a live vaccine known as TY21A. It is a mutant strain of previous vaccine. Weekend can be given only to people 6 years or older because of a live bacteria. Of course, it is contraindicated for younger kids. They can be infected with that vaccine even if it is a mutant strain. Vaccine after injection of inactivated vaccine, immunity after vaccination is much stronger. In the case of parenteral vaccines, it will become for about 2 years, and one only dose is required. A live vaccine should be given as 4 doses, and immunity will appear about a week after intake of that vaccine. And if it is required, if it is citizens of endemic area or constant laboratory worker, then booster dose is required every 2 years for injectable vaccine and every 5 years for oral vaccine. And of course, oral vaccines are preferred because routine road of infection is oral, and local immunity in intestine will not appear if it is injectable vaccine. WHO states that they have 2 more types of vaccine. It is subunit vaccine, which again is based on VI antigen. It is a capsular polysaccharide vaccine known as VI-CPS. It is parenteral and contains the same, or based on the same strain, TY2. And we have conjugate vaccines, which are combined with some other vaccines. The most popular one, registered and licensed in India only, it is a combination of tetanus toxoid, which is acting as a carrier protein for actually the typhoid compound, and provides combined immunity against tetanus and against typhoid as well. But again, vaccine available only in India. We have multivalent vaccines. Again, the most popular one is a combination of hepatitis A and typhoid. It is parenteral vaccines. The same strain TY2 is used, and that vaccine is available in areas where hygienic levels are very low, and both diseases, typhoid and hepatitis A, are of clinical significance. We have new type of vaccine which is synthetic. It means that it is obtained not from bacteria by destruction or by other methods. It is synthesis of artificial compounds of VI antigen. That vaccine should be much more safe because it is absolutely pure and contain only required component. But that vaccine is only in research stage, and some clinical trials are scheduled, but vaccine is not available for medical use. So for today that's all. Thank you. Bye bye. See you later.